Alright, so this is going to be a quick video about making recombinant protein in eukaryotic cells. So first we purify the cDNA from a library, from the genome library. So we cut the cDNA out from the vector with restriction enzymes. And then we ligate that cDNA that's encoding for the protein we're interested in, and we put that um, into a vector. So you cut the cDNA with the same restriction enzyme as the cut you made in the vector that you're going to use to have it expressed in the eukaryotic cells. So first what you do is you transform E. coli with the vector, and then you select the right E. coli cells using ampicillin resistance, and you replicate the E. coli a whole bunch of times. So replicate the plasmid as well. The plasmid's containing your gene that's going to code for your protein that you're interested in, and um, then you take the plasmid from the E. coli cells, and then you use eukaryotic cells and put the plasmid in there, and then you also have a neomycin site on the vector. So neomycin is going to kill eukaryotic cells, and the eukaryotic cells that have this plasmid will be resistant to it because it's making neomycin resistance. And then neomycin is a prokaryotic gene, and then you'll be using eukaryotes to express a prokaryote gene, neomycin resistance. So you're going to need eukaryotic promoters for the neomycin gene and a poly-A site for the neomycin gene. And so the poly-A tail is SV40 for neomycin, and the gene you're interested in is going to be promoted by the CMV part. You'll see that in the next picture. The poly-A tail is being added by the BGH gene. So here's a picture. So this is the promoter for the gene of interest. That's your gene of interest here. It's going to cut here with these different restriction enzymes. And then you're going to have these two proteins that are going to help you isolate the protein once it's made. You have the MYC epitope and the 6' cysteine. So the MYC epitope is antigen, and it's going to bind to an antibody. So you're going to use affinity chromatography to choose for the MYC epitope, and the 6' cysteine works the same as the MYC epitope. It's acts as an antigen, and the term is just a termination codon, UAG or UGA or UAA termination codon. So you have a AUG start codon here, and this F1 origin, you don't have to worry about what that is. And over here, most likely you have a promoter for the neomycin, and poly-A tail for the neomycin, and then you have the origination. So, and for E. coli, it's gonna have to replicate this plasmid, so it needs like a place of origination of replication here. And then this is ampicillin resistant gene that is going to be useful for E. coli. I had a couple questions about this. I'm not sure when I'm going to figure it out. Um, why do you have to put the plasmid in eukaryotic cell to make the protein, as opposed to just having it in just make the protein in a prokaryote? Maybe because the correct folding can occur in a eukaryote. And another question was ampicillin resistance is not transcribed in eukaryotes because no promoter region rather than, so there's no promoter region for ampicillin. Rather, there's going to be a Shine-Delgarno sequence, I think. I'm not sure. So, is that right? And ampicillin resistance should not have a poly A tail because E. coli is using it. And this, it doesn't say so specifically here, but so this should be a promoter site for the neomycin spot.